for being here. So it's my utmost pleasure to welcome Professor Ioannidis here in our talk series. And I think that most of you know some of John's amazing work, only some, because it's really difficult to know all of it, as he has published so many papers that I find it close to impossible to track all his publications. And so he has an extremely wide-ranging and impactful publication profile. His H-index today stands at 222 in Google, as far as I've seen. And his work is getting cited at 6,500 citations rate per month, which is a fantastic thing. And his wide-ranging work is really well exemplified by his very diverse affiliations at Stanford University. He is a professor of medicine, of epidemiology, of population health, of statistics, biocourtesy, and biomedical data science as well. I think that tells a lot about his extremely wide-ranging scientific activities. And I would give the word to you now.
Thank you, Dennis. Thank you for the very kind invitation. I wish I could be at Cambridge today. I love Cambridge and I have lots of friends and wonderful colleagues there. So let me share my screen. Can you see it now?
Yes, I can see it.
Okay. Okay. So I will try to discuss issues surrounding reproducibility and transparency indicators and try to give some evidence on where we stand at the moment and how things have evolved across diverse scientific fields. Some preemptive comments. Science is the best thing that can happen to humans. Science depends on trust. Trust depends on transparency. Trust, sorry for the typo, is enhanced by reproducibility. And the question is, how much trust can we have? Because we really want to have trust to be able to build on what we read, on what we learn, on what we see. We want to make sure that we have some solid foundations. If we take a look at the global landscape of research, we see that there's a lot of darkness in some of the transparency indicators. For example, if you look at the open access papers, this is some exercise that we published with Steli Sergiu from my team and other colleagues from Metrics for indicators like data sharing for the last 30 years in PubMed, which covers the entire biomedical knowledge and also some other fields. There's lots of darkness around the world. And if you ask for registered protocols, again, for the last 30 years, from 1990 to 2020, there's still a lot of darkness. In fact, the little light that there is mostly in some African countries. And this may sound like a paradox, but actually the research that is done there, mostly with support from European countries and high-income countries, has high standards, and very often they have to have a protocol registered and also make sure that they share their data for the important questions that they're asking. But nevertheless, the vast majority of research, you have to take it for granted that the data are what is presented to be, and that the protocol is what was intended to be. So what is reproducibility? Many people talk about reproducibility. In time scale, we see that the terminology is being adopted by a geometrically growing number of papers across all scientific fields. So more and more papers mention that term. But in fact, there's many differences in how different fields are thinking about reproducibility. And maybe these are justified. So there's differences in the degree of determinism, the signal to measurement error ratio. Fields have very different determinism and very different signal to measurement error ratios. The complexity of the designs and measurement tools, many fields just use 2x2 tables and others use very complex artificial intelligence methods. The closeness of fit between hypothesis and experimental design data that would make someone happy. In some areas in physics, if you're off in the 6th decimal point, then the theory is considered to be refuted. Conversely, in some areas in biomedicine, we're happy even having the first decimal or even just the first digit being completely off. Statistical analytic methods to test hypotheses vary a lot between frequentist and Bayesian methods and how these are implemented. What is the typical heterogeneity of experimental results, and what do people think about heterogeneity? Some fields hate heterogeneity. They say that that's not a good sign. It means that there's something wrong here. Others are very happy to see heterogeneity because they can say that now we can explore moderators and kind of synthesize the evidence better and understand why results are different. There's great differences in the culture of replication, whether replication is considered a sine qua non. For example, genetic epidemiology, if you cannot replicate something in multiple studies, you cannot publish it. You cannot publish it for the first time. In many other fields, replication is seen as me too research, very low priority. Actually, people would be chastised to spend their time on replication. Why are you doing this? You don't have any better ideas. You have no ideas of your own. Whether transparency is something that people wish to have or not. About 8 years ago, I was the first to write a paper against Theranos, the most prominent startup in Silicon Valley with 9 billion valuation, and the favorite child of everyone, and the way to revolutionize health. And I just said I see that what they have is stealth research. They haven't published anything, and I don't know if their valuation should be $9 billion or $9. So obviously that didn't go well. But some areas believe in stealth. They believe that you should hide everything. So transparency is a bad thing. And others believe that transparency is exactly what we need. Well, there's also a culture of accumulating knowledge, whether we perform, for example, meta-analysis. Some fields perform 50 meta-analysis on the same question. In some cases in medicine, I've seen that. And in others, there's no meta-analysis in the entire field. Statistical criteria for truth claims. Even if people use, for example, null hypothesis significance testing, some fields choose a p-value of 0.05, others go for 0.005, others go for some adjustment with Bonferroni, others go for 10 to the minus 8 or 10 to the minus 9, and so forth. And finally, why are we doing this research? What are the purposes to which the findings will be put and the consequences of the false conclusions? It could be that it's just curiosity, or it could be that it's applied and it may have major consequences for health, for policy, for public health, for the future of our world. Basically, there's 3 different types of reproducibility: the reproducibility of methods, reproducibility of results, and reproducibility of inferences. Reproducibility of methods means that we are able to understand what is in the method section. This means that we can put this machine of that research paper to work again. We can read the instructions and reconstruct it, make it function again. And we understand the experimental procedures, the computational procedures. We have the algorithms, the codes, everything that is necessary to put it in place. Then reproducibility of results means that we do yet another study, a different study, and we see that the results are corroborating the results of the early study if we follow the same experimental methods. And same can be debated. Should it be exactly the same? Should it be slightly different? How much different? Do we want exact replication, or do we want to diversify? And then reproducibility of inferences is the making of knowledge claims of similar strength from some study results or some number of studies with results. So you give to a number of experts who are knowledgeable, who are well-trained, the same evidence, the same results from one or multiple studies, and you tell them, so what do you make of that? And some of them say, well, we know for sure. Others will say, no, we don't really know for sure. Others will say, we have no clue. Such extreme opinions, of course, are not necessarily very common, but we do see them. And it's very common to see some difference of opinion on what the evidence means, let alone what would be the action steps if this evidence is to decide some action. Statistical methods can be used across very different fields, and some of them are extremely popular, and they're at the epicenter of how we decide about what we have found and what we have reproduced. The classic approach has been frequentist for most of biomedical research and for most other fields. Typically, this is using p-values and also linked to some null hypothesis significance testing to claim significant results. This is a text mining evaluation that I did with David Sholarias and other colleagues, looking at the entire biomedical literature in PubMed. There's about 14 million papers in terms of abstracts that we could text mine, and close to 1 million full text papers. And we saw that when p-values were available in the abstract and or in the full text, 96% of the time, there were p-values that would be coined as statistically significant. So, 0.05 or less. And this has gone down slightly over the years from 98% to 95%. So there's a little bit more room for non-significant results, but it's almost ubiquitous. So that machine is not really very helpful because everything is statistically significant, more or less. So if that was a tool to differentiate what has a stronger signal versus a weaker signal, it doesn't seem to function very efficiently. Along with Dennis, we have looked at a number of different fields. Dennis has put together some amazing tools to probe literatures in large scale, and this is looking at journals of neuroscience and psychology and medical journals. The common feature is that most studies that are published, even in the best journals, are very small. So the power for these studies to detect what would be meaningful effects is very small. And that means that we have tons of false negatives, perhaps, but probably even more false positives if we have even a tiny amount of bias. This is some other work that Dennis has led, looking at Nature Neuroscience, the Journal of Neuroscience, Neuroimage, and Cerebral Cortex, and this is 147 studies. You can see the change in the median sample size over time, and this is getting better. It is increasing. So the regression has a value of barely 5 in the 1990s, and it goes up to something like 25 currently. So there's clear improvement, but even 25 is probably a very small number if you think of what might be the size of the effects that might be chased in many of these evaluations with fMRI studies. This is not just in one field. It's not just neuroimaging. This is a paper where, along with colleagues, we put together evidence across very different types of designs, ranging from basic science, animal studies, translational studies, clinical trials in neuroscience at large. And the common denominator was that the sample sizes were very small. We had power failure. We had very little power to detect effects of even modest size across practically all of these disciplines that are very different, even though all of them belong to neuroscience. And this is not just one scientific field. You can go to a completely different area of the universe of science. You can go to economics. Along with Tom Stanley and Christo Kouliagos, we probed 130 economics topics with more than 70,000 effect estimates, and we saw pretty much the same pattern as neuroscience. This is mostly observational economics, not randomized trials economics, and we saw that in most of the fields, the vast majority of studies were very small, and typically you could have an entire field, an entire topic that had no power in any of the studies that had been conducted. It was very uncommon to have at least one well-powered study in a topic. One should comment that increasingly in some fields in the last decade or so, we have the inverse problem. We have big data. So instead of having these tiny studies that most of us have been doing, I have done many of them, now we have the capacity to use data sets that are collected very often, even with no research protocol and no research preemptive design. Just they happen to accrue themselves while I'm sleeping. While I'm sleeping, people are still tweeting somewhere, or sending messages, or electronic health records keep growing. Unfortunately, people go to the hospital while I'm dreaming, and next day there's more of these data. And This is a valuable resource, of course, and we can hope to learn from big data. But even though the power problem is corrected, it creates other problems because most of these data have problems of accuracy, of coverage, of biases. It's a whole new world that we need to understand how it functions or malfunctions. There's many efforts to try to understand the replication potential of published studies in disciplines. This is an early synthesis that I put together with several colleagues in epidemiology, looking in candidate gene genetics. That was the dominant way that we were doing genetics until about 15 years ago. There's still a lot of that being done, actually, but until that time, it was really the dominant paradigm. We probably had 100,000 papers published in that way, where people were thinking very hard of one hypothesis, one specific gene that may be related to some disease, and then going after this to see whether indeed it is related. This is pretty much the way that most scientific fields continue to work nowadays. You know, we think very hard about our best shot, and then we try to go after it. So going after that best shot, we came up with these 100,000 papers. But then when we moved to a different era where instead of going after the best shot, we could look at every single gene and all the variation that there was in the genome, we could go back with large scale data and see how many of these best shot approaches, the candidate gene approaches, were validated. The validation rate, the replication rate, was just 1.2%. The vast majority of these signals that were published in Nature and Science and the best journals could not be replicated. You know, we had null results when we had much larger sample sizes and large scale evidence on them. This is an effort that I did with Dan Fanelli and Rodrigo Costas. It was published a few years ago in PNAS, where we looked across every single scientific field that had meta-analysis available. And meta-analysis means that you have the ability to put together data on the same question. Now, the boundaries of what is same can be argued. So some meta-analysis include a bit heterogeneous results, others are more consistent. But the fact that someone put these studies together in the same forest plot means that there's some feeling that they can be examined together. So we tried to look across every single scientific field, across meta-analysis that had been done, whether the pattern of the results across the included studies were consistent with biases. And these biases could include small study effects, where small studies find more prominent results compared to larger studies, gray literature bias, where gray literature may show more prominent results compared to peer-reviewed journal articles, decline effects, where we have a first study showing a major effect and then others showing smaller effects, early extremes, where we see a strong effect, then nothing, and then maybe we see something in the middle. So there's an early tension of refutation because the first result created the opportunity to refute it, especially if it's very prominent and published in a major journal. Citation bias, where people cite mostly the positive results, even though the negatives are equally well done in terms of their study design and conduct. US effect, more pressure in the US to get significant results. Does it mean that we get more wrong results or more extreme results? Industry, does it bias the results? Pressure to publish based on country policy. For example, if your salary depends on how many significant results you find or where you publish your papers. In China, you can increase your salary 10 times if you publish in Nature. Does it make a difference? Career incentives, institutional incentives, productivity of authors, other types of impact of the authors, team size. Is it better to have large teams because then you have people who are checking on you and you're checking on them? Countries, authors, average distance between the participants of a team, and then career level, gender, and research integrity. So we can look at these features of these studies and see whether there's biases or at least the patterns of the results are consistent with the presence of particular bias. And we found that most fields have some or most of these biases, but there was a difference in how strong these biases were in different disciplines. For example, small study effects were very prominent in social sciences. They were almost equally prominent in biomedical sciences. They were not prominent in physical sciences. And maybe physical sciences, they don't have a problem with small studies. They have all these telescopes that are collecting zillions of data points, and what would be a small study for them? Gray literature, again, some difference. Decline effect, not the same across different fields. Early extreme citation bias, use studies, and so forth. So each field has its own peculiarities that we need to take into account to understand the reproducibility profile of that field and what we need to pay attention to. In many cases, the reproducibility efforts are showing that the results seen in early discovery are not completely wrong, but they are much stronger compared to probably what is the best estimate for these effects. This is one example looking at the most highly cited papers in biomarkers research in the biomedical literature. And I'm plotting for you the relative risk in the highly cited study that drove the field forward and the relative risk in the largest study, which may not necessarily be completely accurate, but probably it's a bit closer to the quote unquote truth. You see that all the action is below the diagonal. So the largest study almost always finds smaller effect sizes compared to the highly cited study, but even the largest study does show some effect, even though it's a modest or small effect compared to the very large effects that were seen in many of the highly cited studies. If you look at the distribution of effect sizes in a discipline, you can also get a sense of, one, what is nature doing in that field, and two, what is bias doing in that field. So this is an early effort where we looked at 85,000 meta-analysis, including everything that was in the Cochrane database of systematic reviews and covering any field in biomedicine. And we saw that among these 85,000 meta-analysis, if you want very large treatment effect, an odds ratio above 5, something that has been seen in at least 2 studies with a p-value of less than 0.01 and with no obvious evidence for bias, and to be about mortality, we only found one out of 85,000. However, if you ask, are there single studies that could be small or whatever, or biased or whatever, that have large effects with odds ratios above 5, then we found that more than 10% of the studies had such large odds ratios, but always practically, with extremely rare exceptions, when we were to do yet another study on the same question, the effect size went away, and typically it just completely evaporated. So large effects are seen. Typically, the first time that they're seen, they come from studies with a median sample size of about 10 to 14 events, which is really tiny. But then when we test them again, they just vanish. This is looking at a specific discipline, emergency medicine. And along with Austin Parrish and colleagues working in emergency medicine, we examined 431 meta-analyses, every meta-analysis that we could find that informs that very important field. And I'm showing you here the distribution of the effect sizes in single studies and in meta-analysis in emergency medicine. And the pattern is interesting. You see that there is a heavy tail. And the question is that heavy tail real or is it biased? Most likely it is both. But in my experience and in some probing that we did, it seems that bias is at least as prominent a cause as real effects being at large. Can we dissect some of the biases at the level of reported characteristics of the studies? For example, in randomized trials, we think that it is a good idea to make sure that the randomization was properly done. Also, it is important to have allocation concealment. If there's no allocation concealment, people may think that they're randomizing properly, but actually they will just drive patients to the group that they prefer based on their anticipated prognosis. And also double blinding may be important to kind of remove the ability to manipulate allocations and reading of the results. So we looked in a meta epidemiology analysis, the association between these features as they are reported in different publications and the effect sizes across a number of meta-analyses, looking within the meta-analysis and then within fields. And basically we saw that on average, studies that fail to have these protections, we're estimating larger effect sizes compared to those that had proper randomization, proper allocation, and proper double blinding. It was most prominent for subjective outcomes. it was less prominent for outcome like mortality. But we also saw that it was not consistent across all topics and all meta-analysis. There was very large between topic variability, which means that if you have a bias, an overt bias problem, you may suspect that on average, this will distort your result, and on average, it may make your results more prominent, but you're not sure whether it will make them far more prominent, or in some cases, it may make them even less prominent. So when we see bias, we know that there is probably a problem, but we don't know exactly how big and even what direction the problem might take. Increasingly, research is done in a global scale. It is no longer done like when science began in Western Europe, just by a few dilettanti aristocrats who had plenty of money and time and a castle to equip with their experimental devices. And it is no longer done in a few select institutions like Cambridge and Stanford. It is done all over the world. And many locations have scientists who are very diligent and very smart and very interested, but they don't have a strong tradition of running specific types of research. For example, randomized trials and clinical research currently are not done in these epicenters in Western Europe. They're mostly done in other countries. And if you compare the results, and this is what we did in this example, we found that randomized trials done in less developed countries showed far more prominent results compared to studies done in countries with a more established tradition of clinical research. Even for mortality, the difference was startling. And is it that these studies are wrong? I think my suspicion would be that they're not wrong. The ones that are published are okay. But if you do research in a less developed country and you run a small study and you don't find any significant result, that's not going to be published, or it may be disappear in some local journal that is not even indexed in PubMed. Conversely, if you run a small study in a less developed country and you find a big effect, then this has a much better chance of making it to the indexed community of journals. Nowadays, we're facing other challenges. We're facing a challenge, for example, of what I call covertization of research. We published that paper several months ago, looking at how many authors have published on COVID-19. We found 720,000 different authors who have published on COVID-19 papers within just a year and a half that we analyzed. And you can see that they very rapidly populated the entire universe, that cycle of science. And by the end of 2020, the only field among 174 fields that science is divided into that had not any expert publishing on COVID-19 was automobile engineering. And automobile engineering also joined the fray in December 2020. Now, every single scientific field has experts that have published on COVID-19. So that creates new opportunities, but also new drives and new challenges about ensuring that we can have reproducibility and good quality. And this is not easy. There's multiple studies that show that the quality of research on COVID-19 is miserable. And it's far worse than the quality of non-COVID research, which was already bad, including, of course, my own research, which has always been miserable. I'm not saying this to blame anyone. When someone is trying to act with alacrity and produce evidence very quickly, it's very difficult to maintain very high quality. And of course, we have seen things like fraud with papers retracted. This is a classic case of a hydroxychloroquine paper published in The Lancet, the top medical journal, with data from 671 centers around the world. And none of these centers had any clue that this study had happened. The study did not exist. That was egregious. And of course, it was retracted. But there's many other probably situations where it's not so easy to realize that there's a big problem with the data. We also saw a new flourishing paradigm of a shift to models, which had always been there, and they had a share of the literature, but a pretty small share of the literature. Suddenly models became the dominant literature, and they occupied prime places in Nature and Science and drove decisions for the pandemic. Some of these models are very questionable. For example, a classic paper in Nature by the Imperial college team with amazing scientists claimed that lockdowns saved more than 3 million lives in the first wave in Europe. And in this work here that we published in the Journal of Clinical Epidemiology, we looked at the data that Imperial College had used, and we used also another model that the very same team had developed at the very same time, but they had not published in the European data. They had applied it to US data. We saw that other model had much better fit to the data than the one that they had published. And the model with a much better fit actually showed that draconian lockdown had no benefit on saving any lives as compared to other measures, more targeted, like banning large events and so forth. This is a paper that I published recently in Mathematical Biosciences, arguing on whether to improve the reproducibility of modeling, which currently is a large share of the literature, we should think about pre-registration, much as we do for other types of research. And of course, one has to balance the pros and cons. I think that pre-registration is favored when we have rigorous designs that are thought in advance, standardized procedures that are preconceived, optimal choices that are conceived in advance, mostly confirmatory research, outcome, performance evaluation, forecasting, for example, forecasting, all these models that go for forecasting, they can be pre-registered and they can say, this is what we forecast. And there's some very nice examples. For example, the US Forecast Hub and some other efforts in Europe, in Germany and Poland. Projects can be separated into specific steps and data are to be collected prospectively. Conversely, there's other situations where this may be very difficult to do, but even then we should say that it cannot be pre-registered because of these reasons. Going into a bit more depth, if we have raw data, then we can perform not just replication studies, but we can perform reanalysis. We can have a look at repeatability of research findings, and we can see whether we can even get what the original paper did with their own data. And this is one effort that we did a number of years ago, working jointly with Nature Genetics. Nature Genetics was probably the first journal that said that I'm not going to publish any microarray paper unless the authors share their full raw data, their full protocol, their code, their algorithm. Everything needs to be in place so that someone can use them and reuse them or do whatever they want with them. And we found that out of 18 papers that top microarray experts and analysts in the world tried to reanalyze, only 2 of them could be repeated. And the reasons for that failure was that very often the data were not available, software was not available, it was homemade and it had disappeared, the methods were unclear, or everything was clear, but the result was very different compared to what had been published. Now, one might argue that things have become better since then. That was the early days that we published that in 2009. I think that there has been progress. There are some empirical assessments that show that sharing of data has gone up in modeling in general from 5% to something like 15 to 50% in some fields, but it's still not really the majority. This is another field. This is randomized controlled trials, another effort to repeat analysis and see whether reanalyzing the results, one gets the same results. Around 37 reanalyses that had been conducted and published, and 35% of the time, the conclusion of the reanalysis was different compared to the original trial. Now, who did these reanalyses? Because the first thought that comes to mind, probably it was the enemies of the original investigators. Maybe there were some rogue analysts who were trying to put to shame their opponents. In fact, these reanalysis papers that reached opposite conclusions to the original were almost always authored by the same authors. So that sounds a bit crazy, but you can think of our incentive structure and reward system. You cannot publish the same result twice. So if someone reanalyzes their own trial a second time and they find the same result, who is going to publish that? I spent another month and I found exactly the same thing on my data. Good for me. Conversely, if they can build a story by maybe tweaking the analysis a little, and manipulating it in a good way, and come up with a new message that, I told you that everyone needed to avoid this treatment, but now I have found that this subgroup or whatever should be treated and actually should be given priority, then this can be published. And the message now is completely different. This is a classic paper. Psychologists came together to probe the reproducibility of psychological science. I'm sure that you're very familiar with the results, original studies and replications. There can be debate forever on what is the replication rate. I've seen at least 100 different ways to define replication, but they don't look the same. I mean, I think that that's probably the common message. In many, many cases, the results look pretty different. And how about if one reads just Nature and Science, the best journals, very rigorous peer review, everyone is looking at the papers very carefully, not really much better. On average, the effect size in the reproducibility check is about half of the original effect size. Can we predict that? Very nice work by Anna Dreber and her team. Maybe to some extent in some fields with prediction markets, we can tell a little bit better than just chance what is going to replicate. So if you ask experts, do you think this will replicate or not? They will perform a little bit better than chance. If you ask them to play in a prediction market where they invest money and they lose or win, depending on whether they got it right or wrong, they can do even a little better. It's still not perfect. It's very far from perfect. It's something like 70% instead of 50%. And I would argue that probably this is an optimistic result because this is a field where private knowledge is very likely to have. So there may be other studies, there may be studies that investigators have heard about, they're not published, there may be some that have been done even in their own lab, so they know, oh no, I tried that and it doesn't work. Conversely, in many other fields, especially in agnostic research, where nobody has a clue about anything, and you know, it's not that you can do a study in your team. I mean, it's a very large enterprise. I bet that even prediction markets would not be able to function well. This has led to what I call the reproducibility wars. Reproducibility is fascinating. We all want to have reproducibility results that agree with what we have done, but obviously we hate to see that what we have done has failed to be reproduced. In the early days, there were connotations that if that happens, someone is a bad scientist, it's unethical, there's fraud or whatever. And you know, the mere reality probably was that most of what we do cannot be reproduced. So it's not an issue that one should be ashamed. I would be very happy to see that something that I have done is not replicated. So then I can think, you know, what is going on here and try to fix that error or bias or lack of consideration that I had in my early design. But it has been an early growth and growing pains in trying to go through the reproducibility wars. There's tremendous resistance to refutation in the scientific literature. This is an early paper that I published in 2007 with Athena Tatsionis and Nikos Bonitsis. We looked at some topics in medicine that had been as squarely refuted as it can ever be. So for example, beta-carotene is the best chemoprevention for cancer. So you can avoid cancer and you can live longer just by eating carrots. Refuted in 6 very large randomized trials. Absolutely no benefit. There's absolutely no doubt about it. But studies continued to support that claim and to cite the original paper. Actually, they continue to cite that paper more than other papers published in the same journal, Nature, in the same year. And if you looked at how they were citing it in a way that it seemed that that was still true, not even mention of the refuting evidence. In some other cases, the refuting evidence was mentioned in other topics, but it was disclaimed that, you know, these randomized trials, they were different in many different regards. Recently, we looked at that similar question in 4 case studies from psychology and trying to see the balance between belief correction and belief perpetuation, citation bias versus citation balance, where you cite the new refutation evidence, and explicit defense versus absent defense. So, you know, you do cite the new study or studies that contradict the original finding and discuss why you still believe the original versus you completely ignore that they exist. And we looked at ego depletion, facial feedback, money priming, and flag priming studies that had been assessed with, as you can see here, the original sample size was pretty small in all of these cases, 26 to 92, while the replication sample size was typically many, many thousands, you know, numbers from 2,000 to 6,333. And the original effect size is huge in all cases. replication effect size is 0.04, 0.04, 0.03, 0.001, 0.01. I mean, as zero can be. Here's how it looks like. These papers continue to be very heavily cited, and their citation rates have not gone down. Maybe a little bit, but not really in any very substantial way. And the citations are favorable citations. If you look at how many of those are even citing the replication, the vast majority do not even cite the replication, many years after the replication becoming available. So it seems that somehow there's an organized field with believers, and these believers will continue to publish papers on that specific organized field. And, replicators who fail to replicate, they did their job, maybe now they go away. It was a bad dream, and, business can continue as usual. More sharing. Can we do more sharing than we do? We should. A couple of years ago, we created some trouble with that paper that we published in Nature. We got lots of Rotten Tomatoes. We said that all publicly funded genomic data should be made publicly available immediately, without even those who produce them having any chance of looking at them first. Because if they have a chance to look at them first, they have just a tiny share of the data, small sample size compared to the totality that is much larger. So most likely they will produce very reproducible results. Conversely, we need to give credit to everyone, so they will be co-authors and get full credit, but that we should look at the complete picture and make these data readily available. How many people are willing to share data? Along with Tom Hardwick, we looked at the most highly cited papers in psychiatry and psychology over 10 years. And we reached out to the corresponding authors. We told them, you know what? We are willing to put the work, the money, everything. We don't want anything from you. Just send us your raw data and we'll curate them and we will make them available for everyone. You have published one of the most influential papers in the field in the last 10 years, and we want to make sure that the community will have access to enjoy that. And we didn't get that many data sets. Some data sets were already existing in repositories for public use. Some investigators did send their data, but the vast majority did not. And the reasons were not what I thought. I was thinking that the reason would be, well, we're still working on them and we don't want to share because we're still publishing, and why should others publish with our own data? That was not really the reason. Most of the problem was that the data were outside of researchers' control. In many cases, the researchers had not even seen the data that they put their name as first or senior author. Typically with industry-funded research like clinical trials, it is the industry that is running the trials, analyzing the data, writing the manuscript in many cases and sending it to famous professors to put their name on and their prestige. And they don't have control of the data. Then, of course, there's legal and ethical issues. If you have not anticipated that in the informed consent, some people were preparing their own data system. Data no longer existing in some cases. The classic example is a paper in The Lancet in the 1990s. Very curious design and very curious results. So people asked, where's the data? We need to look at them. And the reply was the study had been done in India. The data has been eaten by termites. So we still have other types of termites nowadays. This is another effort that we try to see what is happening in the top medical journals that have decided for a number of years now to have data sharing statements. So they will not publish a paper unless there's a statement on how are you planning to share the data. So yes, most of these trials published in the last few years had data sharing statements, but fewer were planning to make individual patient data available, and very few had placed them in repositories where you could not access them unless you were approved. If you said, okay, I want to use them right now, it's only 2 out of 500 trials. So not much that could be used right now. Should we lose our trust? John Carlisle, editor in Anesthesia, probably has lost his trust in much of clinical research, at least. For a number of years as editor, whenever he was getting a randomized controlled trial, he was asking to see the raw data. And that was a precondition. I'm not going to move forward with the paper unless you send me the raw data. So he looked at the raw data, and in about 30 to 40% of these trials, the data were so impossible that he called these trials zombie trials. Either fraud or complete mess up, like the same exact data in one folder and in another folder, that this could not be. So I published that editorial based on what he published, and I estimated that if what is happening in anesthesia, which is a very respectable journal, probably much better than the average journal in medicine, applies to all medical fields, then probably we have somewhere between 200,000 and 500,000 zombie randomized trials that circulate among us in the published literature. And I published that editorial on a Halloween day, but it was not a joke. Can we do better? Some journals are doing better. For example, BMJ and PLOS Medicine, for a number of years, they have a policy that you don't need just a data statement for sharing, but you need to explicitly give away your data to everyone who's asking. And again, this is a precondition to publication. And we did ask these investigators. Florian Odette from my team reached out to them, and we got close to 50%, 46% of these data sets retrieved. We did reanalyze them, and we found that those that were retrieved agreed with what had been published. There were minor deviations, but not such that would make the conclusion be different. So in a sharing environment with transparency and with a policy that is pretty much rigorous, then you can hope to have very reproducible results, at least repeatable results. Let us zoom out across biomedical literature. This is a survey with random sampling of papers from 2000 to 2014. We saw that very little was being shared, and also close to nothing was available as a pre-registered protocol. We repeated that survey in 2015, 2017. We saw some movement. There was a bit of sharing and some protocol registration. And then we repeated that exercise with millions of papers, the entire open access PubMed Central. And we saw that indeed data sharing has gone up from 0% to about 20% over the years, but not getting any better. So you see that in the last 5 years, we're pretty much plateaued at about 20%. Code sharing is still non-existent. If you look at protocol registration, it's a couple of percent, but no better than that. There's some progress. And there's clear progress in disclosure of conflicts of interest, in funding disclosures. Most papers still continue saying that what we do is very novel, even though it may not be. And there's a bit more of papers that claim that what they do is replication. So they're not afraid to say, I'm doing a replication here, but it's still a minority. And this is how it looks in a different view. You can see the improvement over time, but not spectacular. So code sharing overall is just 1.2%, and protocol registration is just 2.6%. This is maps of science that Kevin Boyek has generated for our work. So science is shown as a granular map of disciplines, and you can see the colors that correspond to the main domains. We have mapped here the use of these indicators of reproducibility and openness and transparency. You see that open access at the level of the journal being accessible, the article being accessible, is pretty much widespread. So this is okay, but just being able to access the publication does not mean that you access more than an advertisement, that some research was done. And I'm advertising the research, it's not the raw data. Data sharing is highly concentrated into some fields. Code sharing is very rare, and protocol registration also is very rare. Across journals, you can see that there's very few journals that share data for most of their papers. It's a very tiny minority. There's no journal that shares code for the majority of their papers that they publish. And there's maybe a couple of journals where most of the papers are registered, but the vast majority have very little action. So tremendous heterogeneity in the adoption of these practices across different journals. We keep on working on understanding the diffusion of these reproducibility practices. And I think what I have come to realize is that it's unlikely that we will see major improvements unless our reward system is re-engineered in a way that these things matter. So our reward system is basically focused on productivity, number of papers, number of citations, funding, volume of funding. I think that we need to find ways to pay more attention to quality, reproducibility, sharing, and translational impact for applied research. And I have created that acronym with Moen Khoury, PQRST. It's like an electrocardiogram. Do these principles get adopted by hiring practices in universities or promotion practices? We looked at 146 institutions around the world that had faculties of biomedical sciences. 92 of them had eligible guidelines that we could review on how they hire or promote people. And all of these guidelines focused on traditional metrics or indicators or performance assessments. Very rarely you would have sharing, only 3 institutions, data sharing, only 1 institution, and none mentioned publishing in open access mediums, registering research, and adhering to reporting guidelines. Now, this doesn't mean that these features are not discussed in actual hiring and promotion discussions in academic committees, but they're not in the handbooks of these institutions. And you can see here that applies at all levels, not just for tenure, but even for hiring young investigators at the assistant professor level. Another dimension where we can improve is peer review. So this is a whole topic that could be the subject of an entire talk or multiple talks. If you're interested in peer review, please do come to Chicago in September. I'm one of the directors of the peer review conference on peer review and scientific publication. And if you want to submit something, please do by the end of the month. To conclude, science has grown to an enormous range of scholarship. And currently we have 200 million scholarly articles. We have a large number of systematic reviews, large number of meta-analysis. We can probe lots of data that help us to probe science empirically, to do research on research and understand where we are and where we're heading. Transparency and reproducibility varies widely across fields and across scientific applications. I think that there's plenty of room for improvement, and I believe that the key is the reward system. Reward system incentives are what might matter eventually for improving what really matters. Many thanks to a number of my colleagues who have helped me understand lots of things that I had no clue about over the years. And it's very difficult to thank everyone. I have co-authored papers with, according to Scopus, I found out it was 6,590 people. Goodness, I feel like a complete idiot compared to their wisdom. And many, many thanks to all of you for being here today. I look forward to an interesting discussion.
Thank you very much, John, for this amazing talk. Extremely informative. So if you have questions, then please show yourself and ask. And John will have to leave at 6 in UK time, right? Straight to his class. Link on.
Yeah, I have a quick question. Sorry, I snuck in today. I'm no longer at Cambridge, but I wanted to hear you talk, so I thought I'd sneak in. So I had a question about incentive structure. So I was lucky enough in that my hiring committee at Sussex hired me specifically because they were looking for someone who follows open science practices. Part of my job talk was giving a talk on data sharing. So I was lucky in that, like, these are the things I believe in, and I was able to find a department that wanted to support that. But I also feel that, the incentive structure does make things really difficult for people that do want to follow those practices, because often following them takes more time than doing things, kind of, shortcutting things. So yeah, my hope is that kind of DORA and, things like this is gonna help to kind of force departments maybe to appreciate that more. So my question to you was really, as someone who is senior and someone who has a lot of experience in the world, what is your advice to kind of young researchers who are just starting out in terms of how they can sort of influence their departments or kind of push their departments to kind of take those kinds of things more seriously, so that they can take so that the young researchers can take it seriously without kind of negatively impacting their career?
That's a very crucial question. I think that it much depends on the microenvironment. And I think that for a young researcher, it's very difficult to change the culture of an entire department. I need to acknowledge that. At the same time, having the correct examples or the best examples or the best paradigms can help somehow contaminate the broader environment. And each one of us can contaminate our environment to different lengths and different depths. So I think that in many cases, it's mostly an issue of people not being aware of how things have evolved. So maybe if they get exposed and they hear about it and they see how things work and why that can make things better, they will be very positive about it. I mean, I don't want to think in a paranoid way that there will always be resistance and the senior faculty will always try to push things down. And I think that there are other situations where indeed the environment may not be receptive. And of course, then one has to decide where they want to spend their career and where they want to spend their life. And there may be better places where they may be more receptive. At the same time, we need a little bit of, how should I call it, some modesty and humility that many of the proposed changes are not very well validated. And not all of them will make necessarily things better. There's lots of very interesting ideas floating around. Some of them may make things much better. Some of them may not. Some of them may make things worse. So the goal is not to make our life more difficult. I very often I hear people say, no, you need to do this and this and this and this and this. Say, oh goodness, I mean, if I were to do this, indeed, then I will have no time to do anything else. Or some people say, so no one should publish more than 2 papers a year. Well, then I would have to quit because I'm a maniac and I write papers day and night. I mean, I can't stop. It's a major psychological problem, I guess. But I think we need to balance what are the practices that are best validated, that can be easily adopted, that we have the best evidence that they make things better. And if that's the case, then it's very natural for scientists to adopt them because they know that by adopting them, they will be winners. They will have a competitive advantage compared to others who are not following them. They will be seen as better scientists. They will be accepted in their community among those people who matter as being better. And I think for many of these new sharing and openness and transparency practices, this is already the case. Maybe not everyone will say, yes, you're a better scientist because you're sharing, but those who matter and those who know, I think, do recognize this. And regardless whether it's an early career scientist or a senior investigator, the reputation and the feeling about one's work will be enhanced.
Thanks. That's kind of encouraging because I guess, continuing to do kind of good meta science work is probably going to be really helpful in that. And so that's, yeah, it's encouraging to hear. Thanks.
Some other questions, please. Please, Levan.
Hello, can you hear me?
Yeah.
Yes.
Hi. Okay, here. Hi, thank you very, very much for a wonderful talk. My question was, my apologies if I missed maybe the relevant slide, but so you're identifying a potential mechanism by which to improve the situation that is changing the incentive structure, the reward structure. Is there any data about that, comparing institutions that have various types of incentive structures and therefore the output is different? Like you've compared journals that have different requirements, but what about institutions or maybe funders or something like that?
So there's no randomized evidence on comparisons of institutions that have adopted one reward system versus another. And it's something that I wish we could have, and I wish that we could do, but it's not easy. I mean, you know, many of the logistic issues surrounding performing experiments around reward structures are difficult because you need to have, you know, consensus at a very high level and consent across multiple institutions. Funding agencies, there's more research, but also funders are a bit reluctant to perform experimental studies because it means that people who work very hard to put their applications together, they're told suddenly that, you know, you're part of an experiment, and many would not see that very favorably. So I have tried to talk with many funders. I think that now they do more research compared to what they did in the past. We have much more evidence. Some of them are open to sharing. They have generated data from their offices sometimes, or they have even given data to other investigators. Australia, for example, is pretty open, and there's some very nice empirical studies that have come from Australian funding agencies and investigators who have used these data, for example, in peer review. So it's something that you need to see it on a case-by-case basis. Other entities like journals, many of them have participated in randomized trials. For example, there's a number of randomized trials along different methods of peer review. And we have evidence on how it is to do peer review with blinding versus without blinding, or with the inclusion of statistical consultation, having statistical consultation. There's even some studies that use fake manuscripts, for example, a comparison where you have a manuscript that is identical in the control group, but the experimental group, the manuscript finds significant results, and the control group finds the manuscript has non-significant results, but it's exactly identical otherwise, and it's sent to reviewers. And of course, the positive manuscripts get more commonly accepted than the negative ones. And the explanation, the comments of the reviewers is that the negative manuscripts have very poor quality and poor design, you know, even though the design is exactly the same. So there's ingenious ways to go after experimental evidence with journals or funders and perhaps institutions in the future. And I'm very open. I would welcome suggestions on how we can do more of that, because we do need it. I think that, as I said, many of the suggested improvements may not be improvements at the end of the day, or maybe even detrimental.
Yes, thank you. Even just observational data would be a good start, right? Just like with other aspects that you've shown with journals that ask people to share code and data, you then actually reach out to the researchers, and 50% or something actually responded, right? So it will be, then that's observational, right? That's not randomized control trial or anything. Yeah. So even something like that would be nice to get like a first rough evaluation of the solution that you're proposing, which I agree with. It seems like that's the correct solution, but just how effective it would be. I think the data would be nice to see that. Thank you.
Thank you.
And Mark, please.
Hello, thank you very much. While we're talking about peer review, I've encountered a certain amount of resistance from editors and reviewers to the idea of multiversing. Now, that's a clever name invented by Sarah Stegen and some other Dutch authors for some good work about doing your analysis in various ways to get some kind of a leverage within a study on generalizability, which is also good for reproducibility. But your typical reviewer, in my experience, wants an easy life. They're not very profound. They want a few factoids on which they can exercise their prejudices. They're not prepared to read that extensive web appendix, giving you the consistency between sundry different ways of dichotomizing your variables or whatever. You can do it on almost anything. You shouldn't do it on everything. You have to limit it. But there are some things on which it is informative to do it. But I meet resistance. Would you be prepared to put multiversing, or whatever we want to call it, on the list of quality criteria, and maybe get it mentioned in your upcoming Chicago conference as an aspect of concern in the faltering peer review system?
Excellent comment. And if you have any work on multiversing, please do submit it for the conference. It's a fascinating frontier that is evolving as we speak. I have to say that I came up with something probably identical to what is now called multiversing in the early 2000s. I published some papers on what I still call vibration of effects. You know, that this is my preferred term, basically meaning that you try to analyze some data set in all possible ways, and you try to see what is the range of results that you can get. And we have done work that dissects the sources of the variability with different choices and different types of choices that you can do. And indeed, it is stunning in many cases how variable the results could be depending on what choices are made and how exactly and who is analyzing the data. So I see that as something that I'm fascinated about. I feel it's like a pet concept that I want to promote. Have I been successful in this, or have people who have promoted multiverse equivalents in psychology been successful in making it a mainstream? I have to acknowledge no. I mean, how many papers really do this? Probably still just some experimental sample. But I think we should see more of it, and we will learn more by applying it in a larger scale. Would the peer reviewers be the ones who need to do it? I think that's not going to be easy. Or somehow, you know, would the editors coordinate this? We need to see who is going to do it, because peer review is an unpaid job. Basically, publishing companies, they make about $30 billion per year. They have a net margin of profit that is one of the highest across all industries, something like 30% or more, 40% Elsevier, I believe. And basically, they have all these millions of people who do reviews for no cost, you know, just for free. We know that peer reviews, the average time invested is just a couple of hours. Many peer reviews is probably just a couple of minutes. And I think that our model is very dysfunctional. So, you know, to do a formal multiverse analysis, you would probably need much more effort and a very different system. You would need to give credit for that. You would need to make sure that this is going to be published, for example, as a separate paper that would be respectable. So it is something that needs to be integrated. It can be integrated in multiple ways. We have lots of pre-publication and post-publication peer review. It needs to find its niche for making a change in the system at large scale.
Thanks very much. Well, I've been doing something a bit like it for years, and my students get rather tired of me for ever recommending it. But I think I prefer your term, and I'll write to you about it later. Thank you.
And I see a couple of questions in the chat to you. If you could check, there is one from Rebecca Williams. That's where they start, I think. Then Sharon and Ulrich Müller, Sedgwick.
So Dennis, do you want to choose?
You could just start with Rebecca, probably.
Yeah.
In order of appearance.
So Rebecca says, apologies, my microphone isn't working at the moment, but I wondered if you had any advice on how to begin setting up a research project with the goal of transparency, repeatability. Are there any concrete steps you would recommend taking? So very interesting question, and something that cannot be answered within a few minutes. Meta research and meta epidemiology has grown as a discipline kind of one step at a time and one project at a time. And I have to say that we use very different designs. I think that much like any other research field, they need to be fit for purpose. So the question is what exactly one wants to study and what would be the best way to go about it. I think that there's no formal guide of what is the best design. It will be the same as if saying what is the best design to do science. You know, meta science and meta research is just one other way to look at scientific questions, and all sorts of designs can be applied. Retrospective, prospective, observational, experimental, surveys, case control, nested case controls, you know, multiverse that we mentioned and so forth. So there's no single answer to that question. Any sort of experimental or non-experimental design structure can be used. The question is, what can be done, what is feasible to do, and what will be informative and less biased to do. And here, it depends on the question. There's no single answer, no one size that fits all. I see then, I'm just continuing to read the questions in the chat.
Sharon.
Sharon, yes, apologies with bad connection. What would you recommend to say advice to young, often underfunded researchers who spend much time and energy collecting their own data and who are then reluctant to share it for fear of being trumped or not given sufficient credit by others who may use their data in psychology at least? So yes, I think that someone who only has one project may be feeling a bit more reluctant to share compared to someone who's very rich and has 1,000 projects and can start giving away their wealth. But I would argue that if we feel that sharing is the right thing to do and may lead to better research, better quality, more efficiency, and then I would argue someone who has even less to share should have even more incentives to share, because that would be a way to open to the community, to create new collaborations, to widen their horizon. So by sharing, I think one project can become 5 projects. Conversely, by hiding the data, you know, maybe that project will never materialize because there's not enough critical mass or critical mass of data and or expertise or cross-disciplinarity to be able to materialize it. So bad examples exist, and bad experiences, I can think of zillions where collaborations have not gone well, but I can think of far more where they have gone very well. And I think one needs to be bold and open and try to engage and share and communicate and collaborate and broaden the possibilities. And I think doing that at a young stage is better compared to a senior investigator who maybe is kind of fixated on how they have done things and they don't want to change the recipe of how they have functioned over time. Then the next one is from Ulrich. I'm at a ski resort in Austria. My internet connection is unstable, but I better write. You mentioned the covertization of research in 2000. How do you explain that we got somewhere in terms of vaccine development and our cities of COVID with conventional research in this deluge of poor quality COVID research? I think that I should not be translated as if 1 million people, I mean 720,000 was our estimate until August of 2021. And now probably it's well over 1 million people who have published on COVID-19. We all did this for naught. It would be very depressing if that were the case. We clearly had many success stories. Vaccines were a clear success story. Another major success story was the paradigm of large-scale randomized trials that could be launched, done, finished, published within 3 months and with very solid results. You know, the classic attack against randomized and experimental studies, that especially large ones, was no, they take forever, they take 10 years. Randomized trials are the deathbed of the assistant professor. That was the classic quote. because the assistant professor needs to be promoted to associate professor within 3, 5, 7 years, and randomized trials will take forever, so they will never finish, you will never get it done. And now we saw that this could be done within 3 months and get very solid results that dexamethasone works and hydroxychloroquine does not work, and tocilizumab seems to work, and remdesivir, well, maybe it does, maybe it does not. But so I think we had clear successes, but the volume of papers, close to 500,000 papers on COVID-19. How many of those were high quality and really useful and helpful? Probably a very, very small minority. So you can see it from the good perspective and from the bad perspective. I think both perspectives are true, but I think it gives an opportunity to think about how we can improve our efficiency to an even higher level. Then Francesca, in regards to multiversing, it sounds like something I've heard not to do for fear people would use it for p-hacking. In other words, I've heard pick an analysis in advance and stick to it rather than trying to get a sense of what the data could mean more broadly by exploring it in different ways. Is the way in which multiversing is good that one should do this and then have the honesty to report it and choose a conclusion that is real and beneficial, or have one of the other concepts? So yeah, very interesting question. I think it's an issue of declaring what is going to be done. So for multiversing, I think it's an ideal opportunity to register a protocol and say that we will try all of these possibilities, and this is the space that we will map that includes 1 million different analyses. If you look at all the combinations, and you can explain why you're doing this, why you're thinking of all these possible combinations. You can also think ahead of time that, yes, I will do 1 million analysis, but actually this one is going to be my primary one. And these 1 million will just be mapping the uncertainty, the vibration of effects, the heterogeneity of results that you can get. So that would be crystal clear. And everyone would say that this is the main result, and this is really the vibration around that result. Now, if someone runs these million analyses just to pick one result that would be the best looking and not tell us that this is what they did, I mean, obviously that's problematic. The problem is that in most of the literature where we see one result being presented, we don't know whether that was the only result being pursued or whether it was a million results that were being pursued and just one was reported. And I think that lack of transparency, that disconnect about what was the intention, what was done, and what is reported is creating the problem. Otherwise, running 1 million analysis is perfectly fine. Running a single analysis where you say ahead of time, that's exactly what I'm going to do, and I'm documenting and justifying that I have my reasons that this is exactly what I want to do, and it's fabulous. Then you defend your choices and you say, this is what I believe is best. Does it mean that the published paper will be exactly like that? I have a paper that will be coming out where we did exactly that. We documented in the protocol our choices. And there were lots of choices that had to be made. And the paper went to a peer reviewer that had found something completely different to what we found. And we went through 7 rounds of peer review, each time asking us to kind of data dredge to change things that would try to get the result closer to what he got. We did that again and again and again and again and again. We just still got the same result as we had found. But then our new analysis, when we justify the changes in the protocol, reviewer asked for this, reviewer asked for that, reviewer asked for zillions of things, and this is what we get with all these different options. So transparency, I think, is key to justify either a single analysis is all I'm doing, or a million analysis is all I'm doing, or here's a million analysis, but also the one that I believe is the best fit for purpose for that situation.
And perhaps just a comment, not in the chat, just a comment to this multiversing issue, and also in reaction to what Sharon asked about, the opportunities for young researchers if reanalysis and multiversing would be properly credited, and lots of open data would be available. Actually, it would be great for young researchers who do not yet have their own data, so they could contribute to the literature using the available data.
That's a very important comment, and I have seen that zillions of times, where senior researchers have been telling young investigators that you need to build your own data, and from point one, and that's an uphill battle. I mean, to create your own data set, especially if you're alone or you have a small lab to help you, and you don't even know whether anyone in the lab will help you. Maybe they will just compete against you, and you're all alone. And you're told to go fight against all these giants who have 1,000 times more data than you do. It's a lost battle. I mean, even if you're the smartest person in the world. Conversely, openness and availability of data can make a very young investigator very competitive, because if that person is very smart and has some nice, interesting ideas, these are tools that can be used, and you can use them right away. Of course, sometimes there's misunderstanding about what these data are and how they function. One needs to be familiar with what is being used. But if anything, I think it would help young investigators to bridge that gap that they have when they start.
Exactly. I think so. And just another comment regards funding replication studies, because it seems to be very rare that funders would be willing to fund large-scale applications.
It is a problem, but I think it is kind of discipline specific. So as I said, there's disciplines now that replication is a sine qua non. So you cannot get funded, you cannot get published unless you have replication embedded as a core in what is being done. In genetics, it cannot be funded unless you have replication as the core of the project. In other disciplines, it's still more difficult, and people are not convinced. They're not so exposed. They haven't heard about the benefits. So it's something that is very field specific, and you need to find ways to find environments that are more receptive to the concept. And even though I'm very much in favor of replication, I want to put an asterisk here that I've seen examples that go to the other end, to the other extreme. I mentioned, for example, topics that have 50 meta-analyses. In antidepressants, a few years ago, we published a paper that looked at 185 meta-analyses. And by now, probably there's 500 meta-analyses, more or less on the same topic. So do we need that? I don't think we need that. We need some replication, but there's some situations where replication is very easy. And then people say, well, I will publish another paper if I do that. And that's not good science. I think we need the right balance, the right amount of discovery, right amount of replication.
Exactly. I'm thinking more about, let's say, neuroscience, cognitive neuroscience, where it's expensive to replicate some studies. So you do need dedicated funding, and it may rarely be available at the large scale. Any other questions, perhaps? We still have 4 minutes left. No more questions? All right. Then, everyone. There is one question now. How do you manage such vast amounts of data?
Good question. The answer is, what is vast? I'm asking with another question, because what used to be vast 10 years ago is tiny nowadays. And I think our capacity to handle data has been increasing amazingly. I could never imagine doing some of the analysis that we do nowadays with the tools that we had when I started doing research. So, and I'm sure that when people look at some of the analysis that we do with big data nowadays, they would say that these were tiny snapshots at best. So capacity is not a problem. I think that the tools are there, software, how to handle big data is not an issue. of course, one needs to know the data. And I think that this is a danger in many of the big data sets. If I like what Bradford Hill used to say, that I don't believe something unless I can repeat the calculations on the back of an envelope. And I think this is true. Even if you work with very big data, I always feel more comfortable if I can just dive into them and take a look at a few observations to make sure that I don't have women with prostate cancer in the data set. I've seen that. so you know, just make sure that the data are basically sound and reasonable, and you know, they will not fail some very simple logical queries. I think the problem is when you just analyze data completely blinded to their infrastructure, their provenance, their features, their basic credibility. I think that's a risk. And the bigger the data will become, that the risk will be bigger. So I always feel one needs to familiarize themselves with the data before using them in their large scale magnitude.
Often perhaps it's not even just the size of the data, but the complexity of the analysis, right? Which is
it's multifactorial, and the complexity of the analysis is also so it's a little bit like knowing what kind of a car you have and whether you can put a Lamborghini engine in it. If you have a wooden cart, you know, putting a Lamborghini engine will not be a good idea.
All right, so we reached the end. We let you go as you have to get to your class anyway in this minute. So thank you very much again for this amazing talk and for the discussion. And it was great to see you at Cambridge virtually.